Hello friends, in this video now we are going to start with our next unit, that is amino acids and proteins. So in this first video here, we'll be doing the basics of amino acids. Always before starting any topic, if you do the basics, then it is very easy to understand the complex topic. When this way you make things easy with the basics. So let's get started. First is amino acid name we have to see. See, the name is amino acid. The name is amino acid. When they have given the name, they have not written acid amine, they have written amino acid, because amino group is on left hand side, acid group is on right hand side. See, amino means NH2 group, carboxy acid means carboxy group, COOH. These two are functional groups of amino acids. Now when we join these two groups with the carbon in between and completing the valency with hydrogen here and side chain here, you know this is the structure, common structure of any amino acid. Always amino group is on left hand side, carboxy group is on right hand side. This is the way it is present in proteins, right? That's why whenever I will make this structure of amino acid, I will always make it like this: amino on left, carboxy on right. In books, when you see these structures of amino acids made, then sometimes they'll write amino here, sometimes here, sometimes here. But when we will be making it, we'll be making it very uniform, always amino on left and carboxy on right. Now see the central carbon. The central carbon of amino acid is symmetric or asymmetric. See, all four valencies are occupied by different atoms or group of atoms. So this carbon is asymmetric. I told you asymmetric carbon is responsible for isomerism. We did a general line that whenever a compound has asymmetric carbon, then it will show both optical and structural isomerism. So amino acids will show both optical and structural isomerism, right? So all the amino acids have one asymmetric carbon, right? See here, all amino acids have one asymmetric carbon. This is a general line, but we have exceptions to this general line. Exceptions are glycine. Glycine is having zero asymmetric carbon. All the amino acids have one asymmetric carbon, but there is no isomer for glycine because the carbon is symmetric, right? And two asymmetric carbons are present in isoleucine and threonine. So these isoleucine, threonine, they have two asymmetric carbons. Now learn one more thing here, that isoleucine and threonine, they have two asymmetric carbons in their structure. So it is very difficult to make these two amino acids in the body. So I can say that these two are essential. That is, we have to take them in diet. We cannot make them in body, right? Now tell me the two semi-essential amino acids. You know, they are arginine and histidine. Let's say question is asked which is semi-essential, but now both are given. Arginine and histidine, both are given. So which one will you mark? Which one will you mark? You will mark arginine. So I can say that arginine is more towards essential category as compared to histidine, right? And now if they ask you a question that which amino acid is essential in children but not in adults, then it is histidine. So you can see here that histidine can be synthesized in adults but cannot be synthesized in children. So when we say out of arginine, histidine, which is more towards essential category, we will say arginine is more towards essential category, right? Now, solubility is because of charges. Any compound soluble in water, that solubility is because of charges. Now let's see in case of amino acids. Anything in body is ionized. Even amino acid gets ionized to give negative charge on carboxy group and positive charge on amino group. So the structure becomes like this. Carboxy is negative, amino is positive. Now the net charge on this structure is 0. One negative, one positive, so net charge is 0. This kind of structure is known as zwitterion. Z for 0, Z for zwitterion. It is also known as ampholyte. This kind of structure has no charge. I told you solubility is because of charge. So this kind of structure will be insoluble now. And because it is insoluble, so it will precipitate out of the solution. Now there is a term Pi. Pi is isoelectric pH. Pi is isoelectric pH. It is that pH at which zwitterion will exist. that pH at which zwitterion exists. So we can say that protein at pi of a protein, that protein will precipitate at its pi, because at pi insolubility zwitterion is there. So protein will come out of the solution, that is precipitated out of the solution, right? So when our pH is equal to pi, then there is no charge, so precipitation occurs. At this pH, there is no charge, so precipitation occurs, net charge is 0. But if pH is less than pi, that is acidic pH, at acidic pH, protein exists as positive charge. Protein has positive charge on it. And if pH is more than pi, that is alkaline pH, at alkaline pH, protein is having negative charge on it, right? Let's see the detail how. I'm going to take a deprotonated form of amino acid. Deprotonated means proton is not there on both the functional group, that is carboxy and amino group, right? And obviously deprotonated state will be present in case of alkaline pH. When pH is more, there are no protons. At acidic pH, there are so many protons, right? So we can say at basic or alkaline pH, there are no protons. So amino acid will be deprotonated and the structure will be like this. Carboxy group is also not having proton. Amino group is also not having the extra proton here. So this kind of structure has negative charge on it. So this is what I told you, that at alkaline pH, protein has negative charge. Now we will be adding 2 protons in this amino acid. In this deprotonated amino acid, we'll be adding 2 protons one by one. So when I add the first proton here, this first proton is taken up by amino group and the structure will be carboxy is negative, amino is positive. Now 1 proton is there. Now this kind of structure is zwitterion and here pH is equal to pi, where there is no charge on the structure. So you know precipitation will occur at this structure of amino acid. Now we'll be adding the second proton also, and now the second proton will be taken up by carboxy group and the structure will be now COOH and NH3 positive. This kind of structure, when there are lots of protons here and now in this environment, this is acidic environment now. This is what I told you, that at acidic pH, protein exists as positive charge, right? But be careful with one thing. When we talk of acidic and basic amino acids, then the charges are opposite. Acidic amino acids are negatively charged, example glutamate and aspartate. But basic amino acids, lysine, arginine, they are positively charged, right? So be careful with this thing. Okay. Now next coming to the general basic of amino acid, that all amino acids are L-alpha amino acids. L-alpha amino acids. What that means, let's say I write the name of any amino acid, glycine, alanine. What that means, see, we are not mentioning it as L or D, we are not mentioning it as alpha, beta or gamma. When we are not mentioning, then obviously it is L, obviously it is alpha, right? Because this is the way it is present in proteins. L-alpha amino acid form of amino acid is present in proteins. Now let's understand what is the meaning of L, what is the meaning of alpha. Here in case of amino acid, the special group is amino group. So everything is in relation to amino group. L means amino group is on left hand side, capital L means left. And alpha means alpha carbon has amino group. Alpha carbon has amino group. When we say L is on the amino group is on left hand side, this we have understood that amino group is present on left hand side in the structure. Now what do you mean by alpha carbon? In case of fatty acid, which is alpha carbon, I'm not talking of alpha beta anomerism here. Alpha beta anomerism we have done in carbohydrates. I'm not talking of that. In case of fatty acid, tell me which is alpha carbon. Let's say this is carboxy group. So you know carboxy group carbon is number 1, next is 2, next is 3 and next is 4. So which is alpha here? C2 is always alpha, C3 is always beta and C4 is always gamma, right? So this is alpha carbon. Now what do you mean when we say amino acids are alpha amino acids? Let's see the structure of amino acid again. See, amino acid is carboxy here, carbon, central carbon here, and then amino group here. You know, here we have one H, here we have one side chain. I'm not making that. Let's make it simple. See, can I say this amino acid is a kind of 2-carbon acid? Amino acid is a kind of 2-carbon acid. In this acid, this is C1, this is C2. So, which is alpha? You know, C2 is alpha. So, C2 is alpha, right? Now, out of the total 2 carbons in this amino acid, which carbon has amino group? C2, or alpha carbon has amino group. So, alpha carbon has amino group. That's why amino acids are called alpha amino acids, right? Now, amino acid in protein is always alpha. Amino acid in protein is always alpha. But free amino acid can be alpha also, can be beta also, can be gamma also, right? On the other hand, amino acid in protein is always capital L, but free amino acid may be L, may be D, right? Example of some beta amino acid is beta alanine, which is the catabolic end product of pyrimidines. Example of some D amino acid is D-serine, D-aspartate, which is found in brain, right? This is it with basics of amino acids. Now in next video, we'll be doing amino acids one by one. This is it. Thanks for watching.